Oh, okay. Thank you for the opportunity. First of all, it was a bit surprising to be kind of invited to as a nominee to the election, and my principle has been that it's always good to have kind of candidates whenever there's a democratic process, and it was maybe a bit disappointing to see that there wasn't that many. But regardless, I mean, I've been participating because I'm also super interested in the position at hand, and thank you all for the trust. I hope I can deliver what's expected. So, as with the previous people or officers, I kind of a quick or brief introduction to who I am. So I've been a professor in molecular biology and genetics here at the University of Eastern Finland since February this year. Have been in various positions before that, but kind of getting used to the idea now. And University of Eastern Finland has two campuses, so Kuopio and Joensuu. So if you Google University of Eastern Finland, you will find both. And I'm placed in Joensuu, close to the Siberia border, which makes us actually a pretty interesting place if people are interested in biodiversity in this part of the hemisphere. So whole genomics field is kind of new to me, and I'm still a bit struggling with my comfort zone when it comes to kind of dwelling into it, but I'm slowly learning. So my real background, as with LADA, I'm actually a molecular biologist, but I come from the more biomedical research. I've been doing mostly work on humans and the more traditional model organisms like mice. So since I've been getting back into the proper biology, I've been wanting to integrate kind of this type of toolbox that we have in the biomedical research more into what can be done with biodiversity and non-model organisms. So basically, I actually started originally my studies as an ecologist, but I was a bit finding it boring because it's mostly, at least at the time, was rather descriptive. So was always keen on figuring out mechanisms, and really my interest towards genomics comes from this ambition of expanding more functional studies into non-model organisms, really do experimental stuff to figure out how certain traits, for instance, which genes are affecting the background and so forth. I'm teaching relatively much and broadly, so genetics is really my big field here, but also molecular and cell biology, evolution, and I also just teach basic courses in zoology as well. And I've been involved in ERGA for soon 2 years as a country representative, and then I'm co-chairing the SSP committee. Just as an example on what we have going on in my group regarding kind of biodiversity genomics part of life is, for instance, trying to understand mitonuclear compatibility in speciation. So actually, a bit like LADA, I've been interested in mitochondria basically all my research career, and here kind of outstanding or super interesting questions are compatibilities between mitochondrially encoded genes and nuclearly encoded genes within mitochondrial compartment, and how those kind of, for instance, function in species boundaries when species hybrids are born, and potentially kind of then these hybrid mitochondria result in hybrid breakdown at later stages. So basically using the hybrids, or in our case, hybrid cell lines, to figure out, first of all, identifying incompatibilities between the two genomic compartments, and then from those trying to understand actually the molecular mechanisms of that incompatibility. So that's kind of what we've been doing quite intensively for the last 5 years, but it's not that easy. We have a recent preprint out of this, and it's 50 pages long and actually doesn't conclude anything. So we're still there. But what is a bit more straightforward is that we can identify, for instance, species-specific traits. Like in this example, we identified that there's a single gene difference between kind of metabolism of two species. So in mountain hares and in brown hares, there's basically in mountain hares higher glycerol 3-phosphate shuttle or turnover. And our initial thought was that this is kind of cold adaptation. So basically in mountain hares that are more cold adapted than the brown hares would have then need for better heat production. And this glycerol 3-phosphate shuttle actually produces heat, metabolic heat. But actually that was not the case. So because we have generated a reference genome assembly for both hares, we could actually go and check the gene and experimentally manipulate it in cultured cells to kind of show that it's not related to heat production, but actually what happens is that once you kind of silence the more active glycerol 3-phosphate shuttle in mountain hares, the mountain hare cells start to proliferate faster. And this is actually a species difference between the two species, because the brown hare cells normally proliferate faster than the mountain hare cells. So we actually reverse the proliferation phenotype to brown hares by manipulating a single gene. Still, we don't really understand the mechanisms, which is kind of boring. So don't really have an explanation why now mountain hares would have specifically this type of adaptation. But it's curious kind of to see the link between metabolism and cell cycle control, which is actually something we're wanting to develop further by actually proposing that as a next research proposal for the Research Council of Finland, and we'll see how it goes. But this is kind of super interesting for myself, this type of research in combining our knowledge of the basically cellular adaptations or the animal adaptations, and then utilizing the reference genomes to actually do functional stuff. Bit looking to the future, I've been also engaged in collecting all types of fungi animals as cell culture biobank. So we have reasonably nice representation, mainly mammals of various types as cultured cells, and as based on our experience in the bunnies, we expect that there's actually a lot of different types of traits that we can study and also experimentally address in these various animals. Since I started initially as an ecologist, my main motivation was my kind of deep passion on biodiversity. Also been basically since a kid running around into nature and collecting stuff and keeping them alive in boxes at home and so forth. So I'm having pretty good knowledge on local or Nordic biodiversity, at least, both animals and plants and whatever. And through here, I've been also getting involved quite heavily in not only species diversity, but for instance, the local barcoding initiatives, the FinPol, so Finnish Barcode of Life. And then I've done also some taxonomy on groups that I've been interested. So I've been, for instance, describing these two species as new to science, and one of them is actually reasonably common. So flies are kind of cool, but just to point out that there's still a lot to discover here, even in reasonably species poor north. Doesn't stop there. So because doing science alone would be kind of boring, I've been also doing a lot of other stuff. So been involved in teacher education, textbook counseling, so haven't been writing textbooks, but counseling teachers who write them regarding, for instance, molecular biology and genomics. Been working with Ministry of Environment through projects that they've been funding, but also been involved in red list assessments, and also National Resources Institute of Finland we've been working with, and as well as through my entomological activities, I've been pretty much into interacting with national and local entomologist societies. Then dogs are cool, so I'm also involved in all types of kennel circles, mainly kind of these primitive Spitz dogs. Been utilizing genomics also there and writing breeding program for certain breed. Plus that in my wild past, I've been also a founding member of a NGO working with environment and human rights and whatnot. Been recently active, kind of trying to popularize science, so been getting some award for it, but also some minor roles in TV series, plus then a podcast series on human-nature relationships in the Finnish Broadcasting Corporation. So these are kind of interaction stuff. And job description basically tells that I'm expected to promote application, demonstrate the value of reference genomes, and identifying pursuing opportunities where these genomes can be used, and then the networking and building relationships and helping position ERGA as a key resource and partner in genomic applications. So I think the first part is reasonably straightforward. I mean, it's evident for most researchers when there's genomic data, we can do reverse genetics and learn much more kind of about the biology of the organisms. My experience is that there's reasonably a lot of lack of knowledge on how much this all what comes to genome assembly production has been going forward. It was kind of surprising for me as well when we started to do the whole business, as well as lack of awareness on the ongoing genome project. So still recently I've been meeting with people who were planning to do their own reference genome, and I was pointing out that have you actually checked that there's this reference called GOAT, and they were completely surprised to find that their organism actually already has a reference genome, because I mean it's been, for instance, on the Darwin Tree of Life list and so forth. So clearly, kind of, although we're all into ERGA, there's still a lot of lack of awareness on how fast the whole field is moving. So that's certainly kind of one of the key things that I think we need to work on, and this will also provide then opportunities for promoting application of the genomes in the future. Probably many of you are also aware that annotation remains a really critical issue for many applications. So we have a lot of reference genomes in inverted quarters, but actually if you use those for let's say analyzing your transcriptome data of that organism, you end up with basically non-informative lists of just locus numbers, and that's it. And this is something that probably needs to be worked out with the SAC committee as well as kind of generally. I don't, probably things have moved on since, but there's still a lot of reference genomes which are poorly annotated, and this is really a big obstacle or hurdle for the practical applications. The non-academic stakeholders are kind of a bigger question mark. So first of all, based on, for instance, my experience with entomologists, societies as well as the nature protection groups and kennel circles. People in general are kind of interested in genomics, but there's kind of lack of relevance for instance, with much of the entomologists, it's good enough to have a DNA barcode-based identification of your, I don't know, group of interest, but going into genomics, they first of all don't really understand the benefits or understand first of all the kind of the whole procedure, plus then don't really see the benefits of it. And same applies to many of the other stakeholders. With conservation genetics, it's kind of easier, so we can provide genomes for to be a basis for instance, SNP-based individual identification and population surveys. But again, those are reasonably, at least in the European perspective, maybe easy to mostly actually ongoing things. So if we think of endangered animals, there's really many which are already monitored using genomic tools. But this is certainly that something that can be expanded. Then there's really big topic, which is the using genomes as potential sources for innovation, and this results in, of course, at least some of the times, commercial applications, which is actually what most scientists are, maybe not commercial applications, but applications in general is what most scientists are up to in their research. But then many perhaps feel uncomfortable with the money-making issue, and this is now also for us kind of trying to align the values between commercial applications of genomes, which comes with a lot of controversies, and then actually the practical use of the genomes which we want to promote. Here's an example from a collaborator of mine who has a patent regarding a use of gene from a sea squirt. And back in the days when they were figuring this all out, everything needed to be done the old-fashioned way of cloning cDNA libraries and so forth. But actually nowadays, for instance, this certain sea squirt has been sequenced, and there's a reference genome existing, and it has this really cool gene called alternative oxidase. It's one of the few animals that has it, so this is more common in plants to have. But the plant versions of the same gene don't work in mammalian context. But this one does, because sea squirts are evolutionarily quite close to mammals. And besides alternative oxidase being able to bypass electron transport chain complexes 3 and 4, that means that you get cyanide-resistant respiration, which might become handy if you're into conspiracies. But there's kind of direct therapeutic applications for these genes as well.
Jaakko, just sorry to interrupt, but we're coming close to the hour, so we'll ask you to wrap up if you can. Thanks.
Exactly. So basically this, the networking within the product community, this is a tough question, because the communities will be always more or less exclusive, even if we try to pretend otherwise. So we really need to target the audience that we want to actually communicate with about the reference genomes. It's becoming, this is actually going a bit on Lada's field, because here the challenges are that countries are quite different. There's growing societal polarization that impacts also science. I'm really looking forward for this upcoming stakeholder survey for ideas on on how to take what to take home from there. But basically, put it briefly, I think the main job will be to increase awareness on reference genomes and their importance across the wider field. This will require me working together with social integration officer as well, when we talk about non-scientific audiences. And we need to really discuss implications of this type of work within ERGA. And I see that also media and communications committee have really important job in kind of helping out here. And same as with the developing ERGA knowledge hub. And of course, big thing is the resources to deliver. So if we want to really be an active partner, we should have also resources to work as such. And as we heard, we have a new funding officer, so I'm looking forward to see what she will bring there. And that's it.